never meant to be used as it tends to be used today. We do a single experiment, it's significant, we declare we found something. That wasn't it. He set a really low bar. It may not be obvious why 0.05 is such a low bar, because it seems 95% of the way, to between 0 and 1, but actually the real meaning of it, and it's beyond what we can do today to show you, is the weight of evidence isn't nearly what the 95% would suggest, and it's relates to that question I asked you at the beginning. The number I think of when I see 0.05 in terms of strength of evidence is more like 0.2 or 0.25. It is about 4 to 1, weight of evidence, or 3 to 1 or something like that. Nothing like 95, nothing like 95 to 5, not like 20 to 1, nothing like that. So it's actually fairly weak evidence if you're just talking about barely significant findings. So I actually don't want to go through a lot of these things. I want to sort of cut to the chase. I'll just read you this, which sort of cuts to this issue that it's very backwards reasoning. It says the argument, and this is about using statistics to disprove the null hypothesis, the argument does not seem to accord with what would be the mode of reasoning in ordinary rational discourse. Suppose I said albinos are very rare in human populations. Therefore, if you've taken a random sample of 100 and found in it an albino, the population is not human. I believe the rational retort would be, if the population is not human, what is it? Or with the corpus delicti in front of you, you don't say, here is evidence against the hypothesis that no one is dead. You say, evidently someone has been murdered. So what they're really saying here is, we normally think of evidence as evidence for things. We don't just think in this sort of backward way, this is evidence against the hypothesis that nothing has happened. We talk in very positive ways. What's the probability that the treatment works? But this whole way of doing statistics, which involves only the deductive direction, assuming the null hypothesis and seeing what the probability is, does not give you what you need to know. And again, that's what's gotten us into trouble. I have a lot of other slides here. I'd like to skip over them. I'll just mention what a hypothesis test is, because many of you have heard of it. The hypothesis test says, it doesn't talk about a weight of evidence, which is sort of what a p-value vaguely represents. It says that we should reject and accept hypotheses if something is significant or not. And I want to just read one quote from the scientist who developed this. He said, no test based on the theory of probability can by itself provide any valuable evidence of the truth or falsehood of a hypothesis. So again, they were saying that probability, deductive probability, can't tell us what's true or what's not. He says, but we may look at the purpose of tests from another viewpoint. Without hoping to know whether each separate hypothesis is true or false, we may search for rules to govern our behavior, which, with regard to them, in following which we ensure that in the long run of experience we shall not often be wrong. So hypothesis tests were this technology that was set up to sort of like a quality control procedure, like you would do in a factory where you were testing light bulbs in a box. And you wouldn't care, and you would reject the box if you took a small sample and there were like 2 out of 3 light bulbs defective. You don't care in any one experiment, whether that hypothesis is true or false. You just know if you follow these rules, you won't be wrong too often. And that's okay for quality control operations. You'll have so many boxes with, good boxes with light bulbs, and maybe you reject it by accident. But in the real world, we care about each treatment separately, and we only have 1 or 2 or 3 experiments, and we want to make the right decision. And this technology of sort of having error rates that only work over time has not served us very well. And Fisher actually went crazy. I'll just read this last quote from him. He says, and because he saw his own developments being used in a very mechanistic way, he said, the concept that a scientific worker can regard himself as an inert item in a vast cooperative concern working according to accepted rules and stressing his supposed duty to make a succession of decisions, he didn't like. He said, the natural sciences can only be successfully conducted by responsible and independent thinkers, applying their minds and imaginations to detailed interpretations of observations. The idea that this responsibility could be delegated to a giant computer programmed with decision functions belongs to a fantasy of circles remote from scientific research, and yet that's exactly what we have today. It's called SAS and statistical packages. So he himself, even though he developed this other approach, was very, very dismayed that our conclusions and studies would be driven one way or the other simply by whether the statistics pass some mark. So this just again echoes the point I made before, that he was supporting, that the conclusions are not, they're not false or true based on some significance threshold, but rather they depend on many, many things, including other studies, how well studies are done, and biologic evidence. So let me skip to, I now want to talk a little bit of just about, let me show you how some of these things are reflected in actual discussions that go on today and at the FDA. This was a drug that completely confounded the FDA. And what happened? This was a drug that where it was a heart failure drug, and it was designed to improve the cardiac function, and they designed the key trial not to look at whether people lived or died, but according to a measure of heart failure, a surrogate endpoint like blood pressure, because they didn't think enough people would live or die during the trial because it was too small a trial to reach significance. But they thought it would have a big impact on heart failure. Well, guess what happened? What happened was that it had almost no measurable effect on heart failure, or very little, but it had a huge effect on mortality. But the study wasn't, quote unquote, it wasn't pre-specified as a primary endpoint. And part of this whole philosophy of frequentist philosophy is that you have to define things very rigidly and you can't deviate from protocol. So the FDA found itself in a complete muddle over this, because obviously a drug that affected mortality is very important, but that wasn't what the researchers had said. So these are the quotes from the people on the commission who were trying to interpret this study. He said, what we have to wrestle with is how to interpret p-values for secondary endpoints, that's whether you live or die, secondary, in a trial which was negative for the primary, that was heart failure or heart function. In a trial with a positive endpoint, you haven't spent all the alpha, so that's the type 1 error, this is the hypothesis testing business, on that primary endpoint, so you have some alpha to spend on secondary endpoints, as though you carry this around in your pocket so you can hear my change. So I carry around alpha, like error to spend. It says, in a trial with a negative finding for the primary endpoint, that is, it didn't help heart failure, you don't have any more alpha error to spend on the secondary endpoint. So they couldn't figure out what to do. So here's two cardiologists, and now they're just spiraling down. Now you see complete chaos. What are the p-values needed for those secondary endpoints? Certainly you're not talking 0.05 anymore. You're out of this 0.05 stuff, and I would have thought you'd like to have seen what you thought was significant and what level. What p-value tells you it's there, study after study? See, they don't even have the language to talk about it. They can't even say, what's the chance that this is true? That's what he's talking about, but they don't have the language. They say, okay, what p-value says you'll get that same p-value study after study? It's all scrambled up. And this final comment, what kind of statistical correction would you have to do that survival data given the fact it's not specified? I have no idea how to do that from a mathematical viewpoint. Now the saddest part here is not only are their sort of brains reduced to mush, but these are cardiologists. You want them telling you about cardiology. You want them explaining, well, how could this happen? What's the biology of this? Is this plausible that you could possibly have a mortality effect and have no effect on heart function? Does that really make any sense? That would, that's why they're there. They're not there to spout gibberish about p-values and alphas and errors. So what this story says is that what you know about how things work and about supporting data and about how all the evidence fits together still is relevant. It's not, that wisdom is not surrendered when you come to the statistical calculations. It's highly relevant. The statistics moves you from one point to another, but where you start makes a difference, because if you start from a point, you give me a certain p-value, and if it's for the effect of copper bracelets on arthritis, my belief in that is going to be very different than the same p-value in the same study when we're looking at a new targeted therapy that has a long biologic development for a new cancer, where you have, it works just the way it's supposed to work and the cancer goes away where it never was cured before. Same p-value, totally different inference. So this is the problem that we have with statistics today, is that many folks don't know how to quite put together the numbers in studies together with their own sense of how things work, and also the evidence that went before. So one technology that gets us a little closer, but it doesn't solve the problem, is confidence intervals. And I'm just actually going to go to just want to point out one problem with confidence intervals are the range is an expression of uncertainty around a given estimate. So so here we have relative risk of 1.05. The 95% confidence interval can be quoted from 0.5 to 2. So that says the range of plausible values in some vague way is somewhere, even though we measured a relative risk of 1, is somewhere between 0.5 and 2. Those are the values consistent with the evidence in that study. Or we have a risk difference of -2%, but the values consistent with in that study consistent with that could be true values anywhere from -5% to 1%. So here's examples of confidence intervals. So if 1 represents no effect, you could have a very wide confidence interval that includes 1, that includes very large effects, and often this would just be called non-significant, but that's really a very non-conclusive result. This is a significant result, but it might include small values here. This is null. This is what we would call the minimum important difference. That is, it has to exceed that to be worth doing a particular therapy. That includes, this is also only partially conclusive. This is definitive, that it's in a strongly effective range. This is pretty definitively negative, that is, the range is such that the therapy almost never is unlikely to be effective. And this is a highly precise value where you still don't know whether the effect is basically worth the intervention. So looking at the actual imprecision in the effect measure and thinking hard about whether that merits using the procedure is one way to get away from completely mechanistic thinking. So let me just show you a few examples of how things are written in the literature that could be a bit different if we stress the confidence intervals and the effect sizes versus the p-values. So you might see something like this. Vitamin E supplementation produced a highly significant reduction in lung cancer risk, a vanishingly small p-value. Where if here's another wrong one. Oh, I'm sorry, that's the same one. So the right way to do it could be like this. Vitamin E supplementation produced a small reduction in lung cancer risk, the difference of only 1%, with a confidence interval of 0.8 to 1.2% with that same p-value. So this is looking at the actual measure of effect and saying whether it's large or small, and then saying what's the evidence for that effect. So it's separating the inference into two pieces. If this is true, is it important? And what's the weight of evidence that it might be true? Let's see. DNA methylation of this gene had no effect on survival in lymphoma, p of 0.20, and a right might be the relationship of this methylation and lymphoma survival was inconclusive because the relative risk was 1.8, and the confidence interval went all the way from 0.5 to 5.6, and if it really was 5 or 5.6, it could be highly significant. So that's a non-conclusive finding. And I'll just actually end with this figure. And just emphasize that what we conclude from medical research only starts with what the data say. It's then our job to take that evidence and combine it with other studies and what we think are plausible mechanisms to decide what we believe, and finally to decide how we act based on the consequences of the actions and the probability that we might be wrong or right. And this would be an example of how it's phrased. You might see this: the results were insignificant because of small sample size. Instead of, the evidence for the effect was modest, but we believe the relationship exists because of. So it's an argument. We say it's modest evidence, but we say prior studies gave similar results. They were consistent with a known mechanism, and there were multiple outcomes of the study that all hung together. This would be the opposite. Instead of saying something like this: of the 40 variables examined, only liver cancer was caused by transfusions, p.01. We might say something like this: there was moderate evidence for the relationship between liver cancer and transfusions, but this was not strong enough to make the association highly likely because prior studies had different results. There was no excess of liver cancer in populations with frequent transfusions, and there's no accepted mechanism. So this shows that there's an actual reasoned scientific argument between the numbers that you cite and the conclusions that you then draw. It just doesn't pop out of the numbers. Actually, this is a real study. This is the first study to demonstrate a therapeutic benefit of steroids in chronic fatigue syndrome, p of 0.06. This was in JAMA. Even though the mechanism was not shown, it was inconsistent with prior studies, and all the other endpoints were inconsistent. So this is the example of an argument not made. They think it just popped out of the numbers. So I'll leave you with this quote. This was actually from a student of R.A. Fisher. He said, "What used to be called judgment is now called prejudice. What used to be called prejudice is now called the null hypothesis. It's dangerous nonsense dressed up as a scientific method, and will cause much trouble before it's widely appreciated as such." So he was highly distressed at what had come to be a culture in the medical research literature of calculations completely driving conclusions without reasoned arguments based on prior literature, mechanisms, and a coherence of a whole body of knowledge. So that wisdom that you bring to the table when you either interpret studies or you hear about research is not irrelevant. It doesn't. It's not trumped by the p-values or the confidence intervals. Those are guides to thinking. Those are guides to thinking, but you need to the help of other experts in trying to figure out whether they should determine what you do next. So I'll just leave that there and open the floor to comments or questions. Yes.
Knowing the research and knowing what you know now, back then you were Angela.
Oh, here's a microphone for you. There's a microphone.
Knowing the research and knowing what you know now, and you were Angelina Jolie's father, what would you consult her on? What would you recommend?
I'm sorry, I was whose father?
Angelina Jolie's father?
Oh, jeez. If I was John Voight, well, I would have had a very illustrious career behind me.
What would your advice for her be?
Boy, from what I know, I think she did, first of all, it was a personal choice. So, that's the number one thing. So these things are personal choices informed by probabilities. And that was actually a very good example of when I make this distinction between evidence, belief, and action. So the evidence was that her chance of getting breast cancer were something, I don't know the exact numbers, in the 60 to 85% range, given that she had the BRCA1 genes and that her mother had died very young of breast cancer, and there were other cases of breast cancer in her family. So that number was very high. Whether that's high enough to undergo a prophylactic mastectomy is ultimately a personal choice. And I believe those numbers. Those are pretty well established, and they've been established both by Mendelian genetics and by population-based studies. So those are pretty hard numbers. So, why she chose to do what she did, it's a personal choice. I think it's completely consistent with the evidence. She could have very reasonably, as many women chose, to ride things out and monitor very closely and hope for the best. But I can't really, it's hard for me to make the judgment. But I do think it was based on very good numbers in that case, and she obviously, with other people, made this choice that would improve her. She felt on balance would improve her quality of life, which is really what it's all about. Using the best numbers, using extra advice, and then making choices based on your values and what you assess are the best estimates. But I don't know what else to say.
So if she was your daughter, you would maybe say that.
If she was my daughter, she's my daughter. She still has to make choices that are right for her life. So, I mean, now you're asking on how I would relate to decision making for my daughter. But, I honestly, I don't think actually there's a reason that surgeons aren't supposed to operate on their own kids and things like that. I don't think it's always best to, articulate one's values in relation to very, very personal choices. But I think the important thing there is that it was ultimately a personal choice. And the numbers did not dictate that decision. I mean, that's the other thing that's sort of consistent with what I was saying. There's a gap between those numbers and what she should do. And that gap is what values you put on the various outcomes. And I can't second guess that. And a father, a second person doesn't substitute for the patient's values. So father or not, I wouldn't intercede there. Certainly not on something like that. It's pretty personal. What? You're going way beyond where I'm going to go. I think she has pretty good advice, and she's a bright woman. Any dental-related questions?
I wonder if you could comment as we know, the financial incentive nowadays plays a critical role as far as research and marketing.
Yeah, can you speak a little louder? I'm sorry.
Yes, the role of incentive, financial incentive. Let's assume an experiment is well designed, well controlled, well performed. I wonder if you could comment, especially nowadays, the effect of financial incentive in terms of the effect of validity or objectivity of the data that nowadays we see it quite often.
Yeah. So, of course, nothing I talked about here had really almost anything to do with, how is that happening? There we go. With the reality that, this is all based on an idealized, it's not looking at the literature as a whole. There's absolutely no doubt that all sorts of pressures and incentives, and they're not just financial, although financial loom large, distort the reporting and publication of studies in the medical literature. It's being shown over and over and over again. There have been many societal scientific responses to it. One is in the medical field is that all certainly randomized control studies have to be registered in clinicaltrials.gov. So you can't just choose to publish or not without anybody not knowing that you did the study. And that increasingly, almost every study that is being supported by the NIH and by PCORI has to be registered at inception. So you don't have a choice of, if you choose not to publish or you publish partial results, in some sense it's known because you can go back to the original registration record and see, was a study done and not published, or what did they declare to be their endpoints? So we're beginning to have responses to this, but the answer is financial incentives in in almost every area of medicine has been shown to distort the literature with respect to given, a range of therapies. And we used to turn to meta-analysis and systematic reviews as a cure for that, but companies and other folks have gotten very sophisticated about distorting literature themselves and using that same methodology because they know how it's going to be looked at. And there have been articles documenting this published in the New England Journal and all the top journals in a whole bunch of cases where we were able to get inside information about what studies were published. It's even the case at the FDA that the FDA is in receipt of information on drug harms, with Avandia, the diabetes drug, being one of the signal examples, that are not published in the published articles, and they can't reveal them. They can't reveal the information that they have by law. So I do believe that we have a very serious problem in terms of partial reporting and non-reporting of studies and outcomes. Sometimes it goes to outright falsification, but those are the extreme cases. I don't completely put the fault of it on industry. I think that everybody is under extreme professional pressures to produce results that look good, and that this is a big problem for the literature, and there are widespread efforts to try to diminish these. Of course, declaration of conflict of interest statements is just one, but those don't go very long because everybody is so conflicted, and sometimes in fields every expert is massively conflicted. You'd have no research published at all. So this is a really very serious medical, scientific, social problem that I think we haven't completely solved, and the more potentially lucrative a given technology is, the more it's subject to these pressures. So I don't have there's no single solution because we live in a society where we want innovation to go forward, and many of these technologies actually do provide huge benefit. So trying to figure out what the balance is between benefit and harm, how to distinguish between the ones that are true and the ones that are false in the midst of these pressures is tricky. I don't think that the medical literature is completely and totally deceptive on these issues, but in individual cases it's shown that it has been. So it's quite challenging. I think this affects, from everything I've heard from talking to folks here, this affects dentistry quite a bit. And in the absence of counterbalancing large randomized studies that can sometimes serve as a counterweight, it's very, very difficult to control this. There are many disciplines within medicine that have a lot in similar with what may be the case with the dentistry literature. Orthopedics is one of them. Very few randomized controlled trials, lots and lots of case studies, case series, case reports of devices. Surgery in general often has that. Not a lot of RCTs and lots of people running with successes. So lots and lots and lots of people are working to counter these, but it takes the eternal vigilance and the continued pressure from within the professions and working of honest, I don't want to say honest, but researchers who can resist some of these pressures to get proper evidence out there. So it's a very challenging problem. Yeah.
In the court system, when they're talking about probability and causation.
Yes.
I was going to say, in the court system, when they're talking about probabilities, for example, medical evidence, is an injury, caused? Did an injury was caused by an event, an accident or something? And they speak about the probability. You just have to show that more probable than not, the injury came from this mechanism or this
right.
accident. And they say it has to be greater than 50% or
right.
51%. What about that as a definition of probability? Is that at all?
So the question, did everybody hear that? They said in the legal system, you have to testify to that a particular injury has a 50% probability or greater of causing a specific injury, that a given exposure has a 50. So I give talks to judges and I've testified a lot. that is actually, that's sort of a form of what we call epistemic probability. So here's the problem. Probability that we can all agree on is almost always based on these, group numbers. So, and there are two levels of causation in the legal setting. What's called general causation. Can exposure, does this treatment work in general? Or does this cause harm, on balance in general? And did it cause harm in this particular case? That's called specific causation. Statistics are very good about the general causation problem. Does the treatment work? Does it cause harm? Whatever. But once you go to the single case, did it cause harm in this case, and at what probability, you're completely moving over to what we might call epistemic probability. Traditional statistics group-based is actually, to some extent, silent on that in the individual case. By definition, it can't tell you which person, is going to have it or not. How to translate those group numbers to an individual number, individual causation number, is very tricky, and some would say it can't be done. So it's beyond the realm of what we can talk about here, but it's definitely in the realm of what we call the degree of belief. The degree of belief, my degree of belief that this person had this injury because of that exposure. You have to go beyond just the group numbers that are reported in the trials. And that's the dilemma of all probability, is that how do you go from group-based knowledge, where the probability definitely, we can all see it, to a number that you apply to an individual. By definition, any number that you apply to an individual is a different kind of probability than this group-based probability. And that's the tension of medicine and of dentistry. How do we take these group-based numbers and apply them to the patients in front of us?
You mentioned that the conflict of interest in research and publishing has many different factors,
Yeah.
different causes. When you boil it down, the only cost for this is monetary. The probability of this statement being true is very high, because there's no other factor when you get down to it. And so the question becomes on value. Do you value the health of the individual more, or do you value money more? And how are you going to change that in society? Schools change that?
Well, I don't know that I'm an expert in this field, but they deal with the inherent conflict of interest. So I would say that any fee-for-service physician is in a conflicted state right there. They know that if they order a particular procedure or diagnostic test, they'll make money, and if they do something or talk to a patient for half an hour and advise prevention, they'll make less money. So how do you deal with that? So every physician in every fee-for-service encounter arguably is conflicted. We teach it through professionalism. And you hope that the folks in the profession have absorbed values that allow them to act not just in their own self-interest, but act in a professional way. And those are, the struggle to professionalize any discipline, certainly in healthcare, and to make sure people adhere to the highest edicts of practice, both in clinical practice and in research, is always challenging, but you hope that the quality of people who go into that profession and the things that motivate them are right. But, you're asking sort of a global question of how do we get people to act with integrity when it's in some ways could be in their interest to act, to cheat, lie, cheat, and steal. And that's a more profound question. I'm not a clergyman or philosopher, but I think that the key word there is professionalism. And there are also disincentives. I mean, once you're revealed to have, to have done the wrong thing, to have put in, I was in Baltimore and there was a huge scandal and there are similar scandals in every profession, of a cardiologist who was head of a prestigious department having put in stents, many, many more than were needed, generating millions of dollars a year, spine surgeons who are getting, $1, $2, $3 million a year from companies. When they're revealed, they're actually pretty serious social and professional recriminations. So that is a disincentive. More and more folks are out policing this and trying to discover it. So they know, a lot of companies are now going to be forced to, and are forced to, reveal the amounts of money they pay to clinicians across the board. This will embarrass people too. So there are strong but not determinate forces that are pushing the other way. I think that most people in the health professions are honorable and professional and try to do what's right, but the incentives are strong the other way. Other people here might have more wisdom than I on exactly how you clean things up. But this is the pressure. This is the nature of for-profit healthcare, and it will always be there. But even when money isn't in the picture, there are strong incentives related to professional advancement that make, there are many folks who have falsified research just because, they got more publications, they got promoted, whatever, and I guess you could convert that into money as well. But I don't know what the resistance is other than structural deterrence and also, professional, high ethical standards. And, most of us pass them and a few of us don't.
Thank you you and Dr. Nowzari for contributing to the better values of the profession.
We all try. Probably everybody in this room tries. Any other comments, questions? Gary? And some.
About the Vioxx specifically, if that was a problem with just statistical analysis, or was there some subterfuge with the actual data?
So Vioxx, that was I think that was a multifaceted. So Vioxx, of course, is NSAID, non-steroidal, anti-inflammatory that was shown to increase cardiovascular harms. So I think that the issue with Vioxx is very complicated, and for me the issue was always that it wasn't so much that it caused harm, but that it was the company pushed its prescribing to people for whom it had no benefit. So the tragedy of it is not so much that it went down in flames because it increased cardiovascular benefits, but because there is a subset of folks for which Vioxx is uniquely helpful and whose quality of life with arthritis and with other debilitating rheumatologic disorders is demonstrably improved by having it, even at the risk of higher cardiac risk. And we take all sorts of drugs that have serious risks, and we take it because they have real benefit. So, that was a case where, the company wanted to market it almost like Advil and widen its distribution to folks who really only were having the harm and having no benefit other than over and above what they could have gotten from other drugs. So,
Testing sample too small?
yes. So the market, the population to which Vioxx in the end was marketed was far, far, far higher, larger, and wider than the populations of which it was tested. But that said, they didn't look, and this is a problem with Avandia, they didn't necessarily look for cardiovascular effects when they were first doing it. They never even thought that they occurred, and also those occurred on a different time scale than the pain effects, which occur almost immediately. So you have to design the studies to look, and you have to have post-marketing. You have to have structures available where drugs that are put out in the market where actually can catch. It's not up to the company to figure out whether they're dangerous or not, where you can actually continue to monitor them. And the FDA has only begun literally a few weeks ago to issue guidelines for how they would do this in a proactive way. And they've learned from Vioxx and Avandia and all these others, the catastrophes that can occur if you just let, things go on and be used by millions of people before you detect problems like this. So there was absolutely issues related to suppression of the information. Yes, in some of the studies and delaying its getting out there. But part of the pressures in delaying it was that it was so widely used and was such a blockbuster and brought in so many billions of dollars. It was really a niche drug that was marketed to be used like aspirin, literally like aspirin. That was its problem. So I would argue that if there wasn't as much financially at stake, they would have been more forthcoming about it. And it wouldn't have been as big an issue if it was only being given to that subset of the population that was experiencing real benefit. The problem is, as I said, that its harm was being experienced by millions, its benefit maybe by thousands. And in the end, that's why it got pulled off. So we lost a potentially helpful drug to a small subset of people because of that. Yeah.
I think it's even more complicated than that too, because those event rates are low.
Yeah.
And the prior probability of Vioxx causing heart attack is low, so you're going to need their
Yeah, absolutely.
mechanism. It's really boring.
Yeah. I mean, I think now it's pretty clear, but
But we have a lot of evidence.
but now we have a lot of evidence. Right. So as it was emerging, it wasn't necessarily believed. It was such a shock. Now we have a precedent. So if another NSAID comes on the market or another diabetes drug, we look for cardiovascular effects. But that was the first one. So you're absolutely right. So it took a long time for us to wake up. But still, they had more information that they were owning up to.